So this webinar is on fungal and bacterial leaf spots. So as a brief overview of the webinar, we're going to talk about the project of which this is a component. We'll go through fungal leaf spots, bacterial leaf spots caused by other problems, other factors, and then going through the process to diagnose an unknown leaf spot problem that you may encounter. So the project is a Levy-funded nursery project, and we are writing fact sheets, pest management plans, contingency plans. We have a large diagnostic capacity component of the project where you can send us sick plants or photos of plants initially if you'd like, and we will either say, look, we need to see a sample, or that looks like X, Y, or Z. We are doing webinars, 4 webinars per year, and also 1 workshop in each state and territory each year. There is a biosecurity component to the project where we're supporting the nursery industry, and the information that we're providing mostly is delivered or extended to you via the NGIA newsletter and the state-based NGIs. And they also, the fact sheets, pest management plans, they're on the NGIA website. So firstly, we're going to go and talk about fungal leaf spots. They tend to be the most common leaf spot that you'll encounter in a nursery. We'll talk about their biology, the symptoms they caused. Separately, we'll talk about powdery and downy mildew because they're just a little bit different, and the management of these pathogens. So we are going to do another poll, and this poll is to basically get an idea of where you sit in your ability to discriminate between fungal and bacterial leaf spots. So are you very good? And you have your opinion verified via pathology testing? Or you're good and it's sort of self-diagnosed, you apply the pesticides and the problem goes away? Or is it more, I think I can tell the difference, but sometimes not sure, I know I need more training? That's good to be able to identify that you know that you need more training. Or do you use a consultant who is knowledgeable, and you can tick more than one box if you need to, particularly with the I use a consultant. Okay, so if you can, okay, we're getting a lot of I know I need more training. It's good to be self-aware. Okay, excellent. All right. We'll just close off this webinar in one moment. Don't have to overthink. All right. We shall move to the, sorry, technical difficulty. It's dropped out of my presentation. And I'll get back to the right spot. Here we are. All right, fungal leaf spots. There are a huge number of different pathogens that cause leaf spots across the range of nursery plants that you may grow. So the listed ones here are just a number of them, some of the more common ones. And these leaf spots can be primary pathogens, as in they can cause the actual spot that you're seeing, or sometimes they can actually be secondary pathogens, so they'll infect the plant material after a different pathogen has started something, or they can simply be a saprophyte. That would be where a plant material is dying, and then it will colonize that dead, decaying tissue and produce spores. Sometimes this can then cause primary infection, later down the track, if there's enough inoculum on your dead older leaves, let's say, it may then travel up the plant, but not always. So there is a very wide host range, at least when you consider the width and breadth of the different fungal pathogens that are present in Australia. And there are more and more that we discover. So we certainly do not have a complete knowledge of the types of fungal pathogens that affect the nursery industry. Some of the symptoms that you may see that are fairly typical are these ringed spots. Sometimes they will have a red margin or a yellow margin. This is the red margin being associated with anthocyanin pigmentation. Whoops, sorry, we'll go back. And we'll talk about that anthocyanin production in a moment. You can see sometimes it has more of a brown halo. This is where the fungus is growing into the plant tissue and potentially releasing phytotoxins which discolor the leaf tissue. Other times you may get a leaf dieback where you can see a clear margin between the dying and the living tissue, and the fungus is just growing down and causing that necrosis as it goes along. If you look at this tissue at higher magnification, you may see fungal growth as per the slide on the left. And if you took that fungal growth and stuck it on a slide, then you may well see spores associated with that fungal growth that would help identify the problem. You do have to be careful, or as a diagnostician, we have to be careful because of the secondary and saprophytic species that can be growing on the dead tissue after a primary pathogen has infected the plant. This slide is fairly complex, and the take-home message is that fungal life cycles are complex. Sometimes they can be simple. So this is a particular type of pathogen. Rhizoctonia fits in this category, where sometimes the hyphae are sterile, and it's the only method by which it infects is having that hyphae grow to new plant tissue and infect, colonize the plant tissue and cause a change in that plant tissue, killing it. Other times you get spores or conidia being formed, and there are different types of spores. So your oomycetes being your phytophthora and pythiums, they'll produce zoospores that can swim and to infect new plants. Other types of pathogens have multiple types of spores. So ascospores being the sexual spore, the conidia being an asexual spore, and there are other types of spores as well. So this isn't to say that you need to know all of this for everything. It's more to illustrate that there are different types of spores, and those spores have different abilities to infect plant material and to stay present in the nursery. Some spores will stick around for a very long time. Even in particularly soil-borne pathogens can be present in the ground for decades without a host plant, and then still infect a host plant when it becomes available and the environmental conditions are correct. So when you become aware of a particular pathogen in your nursery, it's worthwhile checking out a little bit, at least, about the biology of these pathogens. Thankfully for us, most leaf spot pathogens have a similar life cycle and also similar, some common threads that manage most fungal leaf spot pathogens. Most fungal leaf spot pathogens require moisture to germinate, whether this be dew or actual leaf wetness, water on the leaves. Oftentimes they are waterborne, and they're spread in planting material and infected crop organic matter. This photo here on the right illustrates how botrytis can hang around. We have a dead leaf in the bottom of the pot. The spores are present, and this is a higher power photo, and they'll stay there quite happily for quite some time, and potentially then can reinfect the crop even when you don't see that there's a problem. So it is very important that you remove the infected crop organic matter. Warm, humid conditions often favour development, and stressed plants, obviously, they have a tendency of being more likely to be damaged and infected by a wide range of pathogens. So growing healthy plants, which is of course everyone's goal, is ideal. In terms of management options, being preventative is the best option. Once you have a fungal leaf spot infecting a leaf, you will always have that damage, even if there aren't active or living spores. If you've killed that colony, that spot will be there, and it's unsightly. So you want to prevent infections from occurring, and the ways you can do that are to lower the humidity in the growing environment, improve ventilation, minimize leaf wetness. If you have overhead irrigation, you want to irrigate at times when your leaves will dry relatively quickly. Different pathogens require different lengths of time to colonize a leaf, and in terms of the amount of time with wetness on that leaf. So if you are dealing with a particular host plant and a particular pathogen, it is worthwhile seeing if there's research on the amount of time required to infect that leaf. Now it's obviously going to vary, particularly if you have different varieties. If you have a whole slew of different varieties, you'll probably find that there are some that are more susceptible than others. So whenever you're reading something online, particularly when it's from overseas, there may be some variation. And what I'd like to point out is that there is a fact sheet available on a number of different leaf spot pathogens, including anthracnose or Colototricum, Alternaria, Phytophthora. There's a nursery paper on Cylindrocladium, and these are all available on the NGIA website. And the easiest way to find them is to go to the NGIA website and actually use their search box and go fact sheets or pest management plans, nursery paper, and then you're more likely to get directly to that nursery paper, because yes, that's the best way. Okay, so we'll move on to powdery and downy mildew. They're a little bit different from the fungal leaf spots that we had previously spoken about. Powdery mildew is an actual fungus, but downy mildew isn't, strictly speaking, a fungus. It's an oomycete, which is in the same group as Pythium and Phytophthora. These are not true fungi. They've been put in a separate group because they have that motile swimming spore. So they are fungal-like, but they're not, strictly speaking, a fungus. Both powdery and downy mildew infect a wide range of host plants. However, each species has a specific group of host plants that they'll infect. They don't infect all host plants. So the powdery mildew that infects cucurbits is different from the powdery mildew that will infect roses or other host plants or capsicum and so on and so forth. And we do have fact sheets available for both powdery and downy mildew. The same concept is as the lots being narrowly host specific is relevant to downy mildew. So first of all, we'll talk about powdery. They tend to be present on the upper leaf. They tend to be more circular. You can see these colonies here. They are roughly circular. Now, with that said, who's seen powdery mildew just absolutely cover their leaves? You can raise your hand. When it gets to that stage, you'll get some deformed growth, particularly when it's on the new growth. it will deform that new growth. So it's not always circular, but the newest colonies tend to be circular. Whereas downy mildew, downy means down, it's on the lower side, it tends to be on the lower side of the leaf, not always. These are generalizations for which there will be exceptions to the rule. Tend to start at least on the lower side of the leaf, and they tend to be more angular. So you may start to see small sort of reddish brown dots on the upper surface of the leaf, but when you turn it over you may well see that fluffy white growth on the bottom, particularly if you look at it under a microscope. In terms of their management, there are some differences, actually some fairly major differences. Powdery mildew is atypical. We had talked about the previous fungal leaf spots. They all needed free moisture to infect the leaf. Powdery mildew is favoured by warm, dry, but overcast conditions. So it prefers to infect the leaf without moisture. In fact, when you put on water, you will reduce infection by powdery mildew. Whereas your downy mildew is more typical of the fungal leaf spots that we had spoken about previously, except that it's not actually a fungus. So cool, wet, humid conditions, leaf wetness on the leaf, they will all promote your downy mildews. So if you limit your overhead irrigation, maintain good airflow, those sorts of practices that we've already spoken about will reduce downy mildew. So it will be important for you, if you suspect that you have a mildew, to work out which one it is. So you can do that either by looking under the microscope or sending it to a diagnostician if you can't work it out from photos to a certain extent and other modes of investigation in the nursery. Okay, so we've talked about the fungal pathogens, fungal leaf spots. Let's hear, are there any questions at this point about any of the fungal leaf spot pathogens? John, do we have questions?
I've got one here, and yep, we have a couple here at the moment. If you can tell that you have a fungal leaf spot, does it really matter what species you're dealing with?
Okay, so you'll probably have realized as we've been going through, it will matter for some things. If you have, let's say, a Phytophthora leaf spot, it could be present, it most likely is present in the soil. Because it is a soil-borne pathogen, it can jump from the top of the growing media onto the lower leaves and then up the plant. So it will be very beneficial to you to work out that you're dealing with Phytophthora and not something else. If you're dealing with a true fungal leaf spot, let's say Alternaria, Coleototricum, Cercospora, some of the more typical true fungal leaf spots, it won't really matter which one you're dealing with there, but you do need to be able to distinguish between that and, let's say, your powdery mildew and your downy mildews, because the management practices are a little bit different. I guess the best advice is to become familiar with the leaf spot pathogens that you deal with on each particular host plant and be able to recognize the different types. Take pictures, put them up in the tea room, have a folder so that you can distinguish between them.
And I guess that leads to the question that I have for you, Andrew,
okay,
with regards to fungicides, so that you really need to know what the leaf spot is in order for the use of the correct fungicides.
That is true. And the next webinar that we're going to give, thank you for that leading question, John. The next webinar we have is on fungicides, particularly in relation to fungal leaf spots. So we'll cover those sorts of questions in the next webinar. However, as a teaser, I suppose you're right, it is helpful because as soon as you know exactly what pathogen it is, you can tap into research which is present in both Australia and overseas, where people may well have tested different fungicides and determined which ones work the best for that particular pathogen. And also so you can be legal. I mean, that's important, unless, well, Victoria has a little more liberal laws, but unless you're if you're not in Victoria, then you want to make sure that you're being legal.
That's it so far.
All right. We will move on to bacterial leaf spots. Bacterial leaf spots and fungal leaf spots can be quite tricky to distinguish from each other at times. So bacterial leaf spots tend to be more angular compared to fungal leaf spots, but not always. They will often have a yellow halo around them, but not always. And by the same token, some spots will look very similar to bacterial leaf spots. Sometimes bacterial leaf spots will cause a soft rot, and well, with that said, some fungal pathogens will also cause a soft rot symptom. Pseudomonas and Xanthomonas are the main genera of bacteria that will affect the nursery industry. There are others in specific host plants that can be a problem. And across the range of ornamental plants that are grown, there are a lot of different bacterial pathogens, and most of your ornamental plants can, from time to time, get a bacterial leaf spot. Thankfully, the modes of spread and preventing these bacterial pathogens is very similar across all of the different pathogens. These are some more examples of the types of symptoms that you may get. So here there is a leaf dieback symptom. You have that nice yellow margin. You could just as easily see this and think, well, that could be a fungal pathogen. And if we saw this picture, we would look at it and go, oh, well, it could be fungal or bacterial. You could do some research online and go, oh, Anthurium does have a bacterial pathogen which causes these sorts of symptoms, and in fact that's what it was. Another example of a soft rot sort of a symptom, water-soaked lesion on this leaf here. One of the main ways that we distinguish your fungal from bacterial pathogens is to look for bacterial ooze. When you see bacterial ooze, most likely there is a bacterial pathogen present. Unfortunately, not all bacterial pathogens produce ooze. And we have a photo here of ooze, because people say, ooze, what are you talking about? In fact, it is an awesome name for it. It describes what it looks like. You can see that's what it does. It oozes out of the plant. So you can see it at a higher magnification. If you stick it on a slide with some water, with a small bit of infected plant material, sometimes you can see a large amount of bacteria come out, and that's what this sort of milky, viscous liquid is here. Other times it could look basically like this, or maybe it have only a slight margin. Other times, yeah, the bacterial pathogens won't even produce ooze at all, and so then you have to be fairly canny as a diagnostician to do a bacterial isolation, knowing that there is a pathogen that causes these symptoms that doesn't always produce ooze consistently. So in terms of how they spread, crop debris, planting material, rain and overhead irrigation all will spread bacteria around your nursery. It can be spread with touch. It is like bacteria in people. If you sneeze on your hand, shake someone's hand, and then they rub their face, they're in trouble. Plants are the same way. If you touch that spot where there's bacteria present around the lesion, and then go and touch another plant that may have open stomata or hydrophodes, you can potentially spread that pathogen. Or if you plant, if you're dealing with seedlings and there's a leaf spot on the leaf, you touch that leaf and then you pot it up into a larger pot, you're maybe a little bit too vigorous, you potentially can then cause a stem canker in the plant. So you have to be careful and have very good hygiene when you're dealing with bacterial pathogens. And you'll see in a crop, who's seen a crop where you have one plant that has a bacterial infection, you go, oh, I need to get to that, but things come up. Totally natural, it does happen at the best of times, or perhaps the worst of times. And then a week later, you've got a whole slew of plants where the overhead irrigation has then spread it to those plants. So you can raise your hand if you've seen that sort of pattern in your nursery. So it's important to remove those plants, particularly for seedlings. If you're dealing with very small seedlings, it's worthwhile just removing that plant, because who wants to, if you sell a plant that has a bacterial infection, thank you, I see some raised hands. If you see that sort of pattern, removing it can definitely help you in the long run. It's also important to be aware that bacterial pathogens can be seed-borne. This can be managed by hot water seed treatments. The seed companies have moved away from heat treating their seeds. This is primarily because it can reduce germination rate. And it's also tricky. You have to get the right temperature, the right length of time for the particular host plant. But it is very important to start with clean planting material. If you start with dirty planting material, really, regardless of whatever pathogen you happen to be dealing with, then you're just going to grow your problem with the plant. Reducing leaf wetness will help. Avoiding damage to plants, as we've discussed. Copper-based fungicides are basically the only registered product, pesticide product, that are registered. They're the only thing registered to protect plants against bacterial infection. And then we have a fact sheet available on bacterial pathogens in the nursery production industry. Okay, do we have questions about bacterial pathogens?
We do, Andrew. First one was, what is the best way to tell the difference between bacterial and fungal infections?
Okay. The necrotic spot margin being angular versus generally being more spherical, circular, that's important. The yellow margin is often present on your bacterial pathogens. There are a lot of if, buts and maybes. The best way is to become familiar with your host plant, look at the literature online, look at the symptoms that they present for each pathogen, and become familiar, and hopefully you don't see them in the nursery, but if you do, hopefully you're monitoring early and you can go, ooh, that looks a little suspicious. I'll send off a picture to my local diagnostician or to us, and we can say, yes, that does look suspicious. We can confirm it if you send us a sample. That's probably a good summary.
And the other one was, what temperatures can you use for treating seeds?
Okay, so roughly speaking, between 52 and 60 degrees is the range which is generally used to treat your seeds and remove pathogens. Oftentimes it's for a half an hour. There's a fair bit of research which goes into those values, and you have to make sure that your hot water bath stays within, plus or minus a couple of degrees. Otherwise, if you go too hot, you're going to kill more seeds, so your germination rate will go right down. If you are too low, of course, then you're not going to have the same level of disinfestation. So you want to make sure you get a good hot water bath, and you want to make sure that if there's no literature for your particular host plant, then you'll probably want to try and test it out in-house.
So I guess if you're looking at it, doing it yourself, you really need to try a small batch and just check the germination afterwards, and hopefully that those plants will, or those seeds will still germinate and produce a sellable plant for you.
That's right.
Okay, that's it so far. Let's go on.
All right, lovely. Okay, so the next source of leaf spot symptoms are caused by other factors, the growing environment. And it is a touchy subject because most nurseries will endeavour to provide the best growing conditions they can for their plants. Now, everyone knows that there are certain plants that like certain growing conditions, and when you can't produce that, you sort of go, oh, I'll just wear that. And that's fine, because we know that we all have logistical restraints. Other times we'll get these sorts of leaf spots that we're about to talk about, and growers can be resistant to the idea that they potentially have caused the problems themselves. So we'll talk about them, and hopefully you can be more educated and take them into account and keep an open mind as much as possible. So there are leaf spots that can be caused by nutrient deficiencies, insect pests. Edema is quite common, and we're going to talk about that in some detail. Anthocyanin production we'll also talk about in some detail, because we see that those edema and anthocyanin symptoms frequently. And we have to tell growers, look, your plant's stressed. Don't know what the source of the stress is, try and fix it. It's an unfortunate story. So over here on your right, these leaf spots, if you look closely, you can see some spots here. This is where a fly has inserted its ovipositor. That's the egg-laying structure. It's damaged that leaf and put an egg in the plant. This leaf here could be an oviposition site, or it could be a sucking leaf, a sucking pest, where the mouthparts have been inserted. They've fed on the plant, injected a saliva, which then causes a phytotoxic reaction around that spot. Then this host plant here on the left, it's an interesting story. We isolated from these spots. We couldn't find anything on multiple occasions. We looked in the literature and found, oh, there is a nutrient deficiency, phosphorus, I believe, that caused symptoms which resembled what we had seen very closely. So in some ways we don't always know with these spots where there's no pathogen present. It can be quite tricky, and it can take work to disentangle the problem and solve it. So first of all, edema. Edema and intumescence, they're a gall-like symptom, a gall being a ball of cells which tend to be grey, brown, beige in colour, as per these spots here. what happens is the cell of the plant leaf as it's growing produces too many cells. The cells themselves blow up, as in they become enlarged with water. This causes the leaf area to bulge out and look like a gall. And different plant species have slightly different symptoms. The structure of the cells is just a little bit different, and so they'll produce slightly different symptoms. And there are different factors which can cause these symptoms in each host plant species. So it does make it difficult to generalize. From the literature, edema can be caused by excess red radiation, poor air quality, nutritional factors, mechanical injury. Effectively, all these things are plant stress, and it can cause a water imbalance in those cells, which then produce the edema. So this is on the left here on the slide that's just coming. On the left, there's a fantastic example of a gall-like symptom, which is there's no pathogen present. And the same here on the right. The structure of the cell has been changed and it's bulged out. Sometimes those galls they'll bulge out at first, and then the cell, like a balloon, it's been popped and it collapses and it will become sunken. So there are a range of symptoms, but we get these types of spots frequently in our samples, and it indicates to us that there is a stress on the plant. And what you'll need to do is identify the source of that stress. So it's quite complicated, and it does require an open mind, examining all of the different factors that may be going on quite independently of the environmental conditions that you're imposing on your plants. Sometimes it's to do with perhaps temperature, and not even just the temperature, but the change in temperature from your nighttime temperature to your daytime temperature. Extremes in temperature, it can be associated with a production of edema, particularly in some seedlings. And you'll find that some host plant varieties are more prone to the development of edema than other varieties. So it does take some vigilance to disentangle the problem and come to a solution for your particular nursery. What you want to do is examine the growing conditions leading up to the damage, and hopefully you can run some tests over time to work out exactly what's going on, or at least have a better indication so that you can work with it and reduce those symptoms. If it's just one particular variety which keeps on getting these symptoms, then of course you want to consider just not growing that variety, and then your problem goes away. The damage to the leaves will always be present, just like with your leaf spot pathogens. Because there is a physical change in the cell structure that can't be repaired, that damage will always be present. Anthocyanin production is a little bit different, okay, and sometimes it is present in association with edema. So you can see here in the center there is this edema gall-like growth, and then on the outside of that there's this red pigmentation. Those cells have produced anthocyanin, and anthocyanin, it's a water-based pigment. It's the thing that makes beetroot red. It's the thing that makes the new growth of roses red. It's a naturally produced pigmentation in plants. Most plants will have that, will produce anthocyanins in association with stress or other growing factors at some point in the life of the plant. What's nice about anthocyanin is that it's a pigment that can be taken away. So as you know, your roses, the new growth is red. It grows, the leaf matures, as it becomes older, that pigmentation is taken away. So in theory, if you remove the plant stress, you can remove that pigmentation from the cells. So the types of factors which can influence a plant to produce your anthocyanins are things like your light, cold temperatures, water stress, and other plant stress.
Muted.
That look at the literature and try and work out what is an association with that pigmentation. What you should also know is the anthocyanin production itself can allow the cells to withstand that stress factor. So in some ways the production of that anthocyanin is good. It helps the plant resist that factor, and it tells you that the plant is stressed. So you can then react. I'd rather see anthocyanin production than any of the other previous things that we've spoken about today, because you can fix it, which is great. When you look at the anthocyanin in the cells at high power, here on the left, you can actually see the individual cells and they're red. So if you've got a microscope and you can go up to, this is probably more around the 80 times, you potentially can diagnose this yourself quite easily. And you can see it, you've got your red pigmentation along the edge of the leaf. Now sometimes the anthocyanin production is in response to infection from a pathogen. And then, well, you're in trouble then, because the anthocyanin is probably going to stick around for as long as the leaf spot's there, which is of course the rest of the life of the leaf. So, but focus on the good thing, you can get rid of it in some instances. Okay, now other leaf spots. We don't go into a lot of detail here on burn. When you see a plant that has a leaf spot, you want to look at it from a holistic point of view. You want to look at the pattern of the damage. And you can look at this picture closely. You've got the leaves about 2 or 3 nodes down, and there's the damage. The older growth is undamaged. The new growth is undamaged. This is painting a picture to us that you're dealing with burn. Or some environmental condition which has imposed the symptoms or caused the damage. But it's in a discrete time period. If you are seeing the new growth being damaged, and the new growth that comes out is still being damaged, you'd be more suspicious that you're dealing either with an ongoing problem or something that's pathogenic. Okay, so it is important to look at the pattern of damage on your plant. And this feeds into diagnosing unknown leaf spots and unknown damage. You want to, first of all, take a hand lens, look at it closely. Can you see anything there? Are there any insects or mites present? Because a hand lens only is so good, times 10, maybe times 20 if you've got a good one. It is worthwhile looking at it underneath the microscope. Are there insects or mites present at higher magnification, at 40, 50 times? This will help you be more confident. Perhaps you can see some fungal structures, some black circular dots or fungal growth. This will help you just to determine what's present. You might be able to see some red pigmentations within the cells, but no actual damage to the surface of the plant leaf. You want to examine these things carefully to produce a story for fixing your plant. What's caused the problem, and then how do you fix it? As we said, you want to look at the pattern. what is the pattern on the single plant and amongst your crop? Is it clumped or scattered? Is it just along the edge of the block? Are the mother stock plants also showing symptoms? We will often get plants and we'll go, okay, well, how are your mother stock plants healthy? And particularly for seedling producers or propagation nurseries, there's a fair bet if your seedlings are infected, your mother stock plants are probably also infected. And there have been a number of occasions where we've gone, okay, can you check your mother stock plants? They go and they check the mother stock plants and go, oh, actually, I didn't notice it before because it was so minor, but some of the plants are starting to show these symptoms. And it helps to get to the bottom of the problem. You want to ask some other types of questions. Are all of the varieties showing symptoms, or is it just some of the varieties? You want to look at the root health of the plant. As we indicated earlier, some leaf pathogens can also cause root rot. And if you have unhealthy roots, regardless of maybe there's no pathogen there, but if you have unhealthy roots, I mean, it goes without saying that your plant isn't going to be as healthy. Perhaps the leaf spot symptoms are simply a symptom of poor root health. And the same, you want to check your stem vascular tissue, the stem on the outside and the vascular tissue underneath, maybe further up, closer to the edge of the canopy, and then also at ground level. Wouldn't necessarily do that if there's only one plant that's affected, but if you've got a crop and you're about to lose it, you want to know, sacrifice a plant, cut it open, look for unhealthy vascular tissue, whether it be green and healthy, or let's say yellow to brown to black, which tends to be unhealthy. So looking at the roots and the vascular tissue, it's sort of like when you go to the doctor and you say, I don't really feel right, I'm not sure what's going on. And as a matter of course, your doctor looks down your throat, looks in your ears, checks your breathing. These are the questions that you ask and you look to do in a similar way. Okay. Then you want to ask yourself, and these are the tough questions here. Did these problems, these symptoms, occur right after a management change? Was it a change in irrigation, a change in the nutritional regime, a pesticide application, or was it an extreme environmental condition? Because plants don't grow overnight, so to speak, it takes some time for those symptoms to grow out, and you've got to backtrack and get to the bottom from the beginning of when the symptoms occurred. And likewise, check your media pH and DC. Now, there have been times we've been with growers and they'll tell us, look, these plants, there's nothing really particularly wrong with them. They're just not vigorous. They're not growing right. And we look at them, look at the leaves, look at the roots, they're all healthy. Look at the vascular tissue, it's healthy. You go, okay, well, what's the growing media like? Have you checked the pH and EC of it lately? No, I haven't. We go and check it, and it's down around pH of 4. Okay, that can paint you a pretty good picture. Then perhaps that particular variety is prone to, or prefers to grow in a slightly higher pH. Fix that, and if you still have your problems, then look further. Okay. So we have a little exam now. We have 4 pathogens, and you'll have to forgive me, I have used this in workshops that you may have been to. If you've been to one of our workshops and seen these pictures, well, hopefully you get 4 marks. There's a Xanthomonas, a bacterial infection, Cercospora, which is sort of a fungal leaf spot. There's downy mildew, which is the oomycete, and there's black spot, which is also a fungal, true fungal leaf pathogen. So the question is, which one is which? So if you would like, you talk amongst yourselves if you have more than one of you, work it out, raise your hand when you're ready to go to the next slide, which has the answers on it. Okay, I've only got one hand raised, so we need some time. Don't overthink it. Uh-oh, John, am I still muted? You sent me a message. Bugger.
I can still hear you, Andrew, but I'm not sure about the rest of the people.
Oh.
Just on the panelist it says you're muted.
One moment.
We've got a couple of raised hands to Andrew's question, but I want to clear you all now. Can everyone just indicate by raising your hands if you can still hear Andrew?
Okay, so...
Muted. Unmuted.
Okay, I'm going to have to just ring back in.
Andrew, you're right now? You seem to be back on track.
Yes, I am. Yes, I am, but I'm also showing a big echo. Just one moment.
Muted.
Ansel knows the answer to the various leaf spots that are on that slide. We'll go back to raising hands again. Has everyone had a chance to have a look at those and work out which is which? So just raise your hands if you have. It just shows the complexity of trying to identify leaf spots. They're not as easy as it may seem. If you are unsure, it's always best to send them to a diagnostician so that they can actually determine for you what is what. And unfortunately, I don't have control of the slide, so over to you.
Thank you, John. I think I'm back. I'm not sure what happened there. We love technology. Sometimes it goes wrong. That's okay. We'll move on. Okay, so hopefully you guys have had time to look at this. We will move to the next slide, which has the answers. Downy mildew up on the top left, your black spot on the bottom right, Cercospora bottom left, and your bacterial xanthomonas on the top right. Now when you look at these, you may see some similarities, let's say between the symptoms here for the downy mildews and the black spot and the Cercospora and the xanthomonas. But when you look at them underneath the microscope at higher power, you'll see much different symptoms, as much different spores. So chalk and cheese, in fact, with the xanthomonas. This is the ooze that we saw with that rose. This is the downy mildew with that tree-like spore structure. The black spot with the sort of figure 8 or human waist line, and the long Cercospora spores. Now we don't expect you to know all these things. We just want to illustrate that your diagnosticians are valuable to you. It's a pat on the back, really. Okay, so we have a poll. And we're going to share that here. How many did you get right? Now, for some reason, just having a technical difficulty. Hide. One moment. How many did you get correct? Thank you. You should see it on the screen now. So, thank you. We're getting a few with 2 correct. Yeah, because of course if you get 1 wrong, then most likely you have another 1 wrong as well. Excellent. All right. We're going to just close that poll and move on because we are coming very close to the end of the workshop. Now I put this one in as not an exam per se, but to illustrate that you get a lot of different spots on a single host plant group. These are cordyline. And so here we've got Phylosticta, which is a fungal leaf spot, Phytophthora, a Phytophthora leaf spot, your virus being similar to the botrytis spores. You've got some abiotic. You can see the anthocyanin production down here. Okay, and insects can even cause some problems, at least in certain areas on the cordylines, in certain geographic regions. Okay, so it's not always simple, and it can be worth sending off your sample. Through Grow Help Australia, the project provides for discounted diagnostics, and I guess this is a bit of a plug. So all production nurseries receive discounted diagnostics. NIASA accredited nurseries receive 10 free samples per year. So that's almost one per month. Hopefully you don't need them all, but they are there so that you can take advantage of our service and manage pests and diseases better. We've had people take the final reports, for which this is an example, and use them to educate their staff. They take the pictures, the close-up pictures of the symptoms, then they can use that with their microscope and go, oh, that looks like that. And they can build a library of different problems that may occur on different plants. And what we'll do is we'll show the types of tests that are present, that we've done. We'll always have the laboratory observations, what we've seen under the microscope, and just by looking at the plant, the different tests that we've completed, some pictures of the symptoms and perhaps close-up images of those symptoms. We'll have a summary of the tests, and then we'll have some recommendations. That is a conclusion. We thank you for attending the webinar. We'll just be sending out a survey in an email with some follow-up links where you can reach Grow Help, the fact sheet links and whatnot. You can email the Grow Help email address. That goes to a number of different people on our team. And we will take final questions now. Any final questions that you have?
Andrew, we have a couple here. What is EC?
EC is electrical conductivity. So it is a measure of the salt's concentration effectively. That's the simple way to put it. And since I'm not a soil or biologist, that's best. So when you have very high salt content, of course that can cause some problems for plants. So you want to make sure that it is within a range. If you have extraordinarily low salt content, it can also cause some problems. So you want to be relatively low, but not too low and not too high.
And the other one was with regards to edema. Even if all the conditions are perfect, why would people still be getting edema in the plants?
All right. So plants sense things differently from people. And it's actually interesting. There's a growing body of research on what plants can sense. For example, there's evidence that plants can hear music. There's evidence that they can, or some plants, I was reading a paper not that long ago, that a plant was dropped. It was a sensitive plant. So the plants that can that close their leaf when they're touched. Okay, and this researcher dropped the plant from a height and then stopped it. And it caused the plant leaves to close. And they did this on a number of occasions. The plant learned that this stimuli was safe. And then it got to the point where it wouldn't open its leaves when it was dropped and stopped rapidly. What's also interesting is they did this, they stopped doing it for a month, and then they did the same test with those plants. They dropped it, and the plant still didn't close its leaves. Obviously, not all plants can do that because they can't all open and close their leaves. What I'm saying is that plants can sense things that we don't give them credit for. And when they're sensing something that we don't sense, it is possible for the environmental conditions or the growing conditions or just whatever happens to be present in a particular time, it's sensing something that we can't work out. And that's why those symptoms occur. The tricky bit for us is to work out what those conditions are.
Okay, and one more question. What magnification do you need in order to look at bacterial ooze under a microscope?
Okay, so you'll need a compound microscope. That is a microscope where you can stick a slide. In general, there are a few bacterial pathogens, let's say bacterial wilt. It goes on a large number of plant species, including your capsicums, tomatoes, other solanaceous crops. If you cut the stem and stick that in water, you will see milky ooze come out of that stem. Unfortunately, most bacterial pathogens aren't that spectacular, and you have to look at it underneath times 10, well, basically times 100 times 200 magnification with a compound microscope. And if you're going to be doing this, I recommend that you get some training to start with, because when you start looking at leaves, little tiny bits of tissue cut up, some plant species have very milky sap, and it can sometimes be difficult to distinguish between sap or just plant bits coming out of a leaf and bacterial ooze. So most of the state departments of ag or primary industries do have some bacterial capacity, and if you want to take some plant pathogen courses at universities, that is another option. But it's worthwhile getting training, and getting a compound microscope is for advanced users. It's for people who have had some training and that are going to be using those skills.
Well, that seems to be all the questions, Andrew.
Excellent. We've gone over time. Sorry about our questions.
I'm just going to thank you from Karen. Thank you very much, Karen.
Yep. So we've gone a little bit over time. We've been some technical difficulties. Sorry about that. But thank you for attending. As I said, an email will be coming out, and there'll be a link to the previous webinars that are already available on the YouTube NGIA channel. So thank you very much for attending. Okay, goodbye.